So coming to the oogenesis. So the oogenesis, it is a process of maturation of primary oocyte to mature ovum, and usually the primordial germ cells, they are present in the cortex of the ovary. Usually these primordial germ cells, they are formed by the proliferation of the endodermal cells of the yolk sac. They migrate to the ovary, they migrate to the gonads at the third week of development, and after entering into the gonads, these primordial germ cells, they undergo mitosis and they form oogonia. These oogonia, at the third week of development, they are transformed into primary oocytes. So usually these primary oocytes, they consist of primary oocyte. This is the primary oocyte surrounded by a layer of follicular cells. Okay, so this is the primary oocyte. So this is the primary oocyte which is surrounded by a layer of follicular cells. So after that, the primary oocyte undergoes meiosis 1. So first of all, the oogonia, they are transformed into primary oocyte by the proliferation of follicular cells. So, and this is the primary oocyte. If you take the primary oocyte, so this is the nucleus, and the follicular cells here, they multiply. First, the primary oocyte is surrounded by a layer of follicular cells, and after that the follicular cells multiply. Follicular cells multiply. And this is called primary oocyte. So the oogonia without cell division, they transformed into primary oocyte. This is the nucleus, and this is the this is the primary oocyte surrounded by the follicular cells. This is called primordial follicle. The primary oocyte which is surrounded by the follicular cells is called primordial follicle. So this is the primary oocyte surrounded by the follicular cells. So first of all, the primordial germ cells, they undergo mitosis and they form oogonia, and the oogonia without transformation, they are without cell division, they are transformed into primary oocyte. The primary oocyte, it is surrounded by a follicular cells. This primary oocyte, this is the primary oocyte. It is surrounded by follicular cells. This is called primordial follicle. So the process of maturation of primordial follicle or primary oocyte to mature ovum, this process we call it as oogenesis. So coming to the primary oocyte, now it undergoes the meiosis 1. It undergoes the prophase of first meiotic division, and after birth only the primary oocyte completes the prophase of first meiotic division, and it passes a dictyostene stage, and it passes a prolonged dictyostene stage, and this primary oocyte, now it undergoes meiosis 1 and gives secondary oocyte. This is the secondary oocyte, and this is the first polar body. Coming to the primary, how the secondary oocyte is formed, you will see to the diagram. So this is the primary oocyte. Now some spaces appear between the follicular cells. Some spaces appear between the follicular cells, and the follicular cells, they are arranged into outer layer and inner layer. The follicular cells, they are arranged into outer layer and inner layer. Outer layer of follicular cells, we call it as stratum granulosum. Outer layer of follicular cells, we call it as stratum granulosum, and inner layer we call it as cumulus ovaricus. Cumulus ovaricus. Outer layer of follicular cells we call them as stratum granulosum, and inner layer of follicular cells we call them as cumulus ovaricus. First of all, the primary oocyte, it is surrounded by a layer of follicular cells. Later on, what happens is the primary oocyte, in the primary oocyte, among the follicular cells, there are some spaces appear in between the follicular cells. Later on, these spaces unite and they divide the follicular cells into cumulus ovaricus and outer stratum granulosum cells. The cumulus ovaricus cells, they immediately surrounds the primary oocyte. Now this primary oocyte, it undergoes mitosis, it completes its meiosis division, meiosis 1 division, it completes its meiosis 1 division, and it gives secondary oocyte. So this is the secondary oocyte, which consists of haploid number of chromosomes, that is 22X, and it is first polar body, because when primary oocyte, when it undergoes meiosis 1, only one cell, it receives major amount of cytoplasm, it becomes the secondary oocyte, and the other cells which receive scanty amount of cytoplasm, it is called first polar body, and further later on the first polar body degenerates. And now the secondary oocyte, it undergoes the second meiosis division. It is nothing but the homotypical division. But this secondary oocyte, it completes the second meiotic division only after fertilization only, and then after fertilization only it turns into mature ovum. If the fertilization doesn't occur, the secondary oocyte does not turn into mature ovum, and further menstrual cycle follows. And coming to the ovulation, ovulation means release of secondary oocyte from the Graafian follicle. We know that, for example, now we will draw the structure of Graafian follicle. So the Graafian, this is secondary oocyte. Okay, and this is secondary oocyte surrounded by layer of follicular cells. And this is a zona pellucida, and here is the first polar body is here, and this is the cumulus ovaricus cells. Cumulus ovaricus cells is there, and outer we have stratum granulosum cells are there. Outer we have stratum granulosum cells. So outer we have stratum granulosum cells are there, and outer to stratum granulosum we have one layer is called tunica. Tunica interna, and above tunica interna we have one more layer is there. So this is tunica externa. Tunica externa, and this layer is tunica interna. These are granulosa cells, and these are cumulus ovaricus cells, which immediately surrounds the secondary oocyte. And this is the first polar body, and this is the first polar body, and the membrane which is surrounding the primary oocyte, it is called zona pellucida. It is a glycoprotein. Zona pellucida, it is made up of glycoprotein, and it consists of small villi-like projections. It gives nutrition to the primary oocyte. So zona pellucida, it is a thin membrane between the cumulus ovaricus cells and the oocyte. So zona pellucida is the membrane made up of glycoproteins. It provides nutrition to the oocyte. Okay, so this is the structure of the mature Graafian follicle. So here you can see the structure of Graafian follicle. So this is the secondary oocyte. So secondary oocyte means it undergoes first meiotic division. So you will able to see only haploid number of chromosomes 22X and first polar body in the perivitelline space. And this is surrounded by the zona pellucida, which is a membrane made up of glycoprotein and consists of numerous microvilli-like projections which provides nutrition to the oocyte. And surrounding the primary oocyte, we have a layer of follicular cells called cumulus ovaricus, and we have a space called liquor folliculi, which is filled with fluid, and the outer layer of cells we call as granulosa cells, and outer to the granulosa cells we have another layer which is formed by the condensation of the fibrous stromal cells of the ovary, that is tunica interna, and outer fibrous layer we call it as tunica externa. Inner vascular layer called tunica interna, outer fibrous layer called tunica externa. This is the structure of the Graafian follicle. At maturation, what happens at ovulation? What happens is this oocyte is released. The secondary oocyte is released, and by the action of the fibrated ciliated epithelium of the uterine tube, it enters, this secondary oocyte enters into the uterine tube. After entering into the uterine tube, if fertilization occurs, then only this secondary oocyte, it completes meiosis II and converts into mature ovum. Just before the fertilization only, the secondary oocyte completes its second meiotic division and converts into mature ovum. If not, secondary oocyte shuts along with the menstrual bleeding. That's about the secondary oocyte and the mature ovum. So oogenesis. Oogenesis means the maturation of the primordial germ cells from primary oocyte to mature ovum. The maturation of the primordial germ cell from primary oocyte to mature ovum is called oogenesis. Usually these primordial germ cells, they are present in the cortex of the ovary, and these primordial germ cells, they undergo mitosis and they form oogonia, because 44 autosomes and remaining 2 are X chromosomes, and these oogonia, they enlarge in size and convert into primary oocyte. The primary oocyte, now it is surrounded by a layer of follicular cells. So here you can see the primary oocyte surrounded by follicular cells, and this primary oocyte now enters into the prophase of first meiotic division, and after birth only this primary oocyte completes the prophase of first meiotic division and converts into secondary oocyte. And secondary oocyte, so now here meiosis I takes place, so the chromosomal number is reduced to half, and after meiosis I, only one of the cell receives the major amount of cytoplasm, and it becomes secondary oocyte, and the other cell which receives the less amount of cytoplasm, it is called first polar body, later it degenerates. And coming to the secondary oocyte, this secondary oocyte is mature at the time of ovulation. So ovulation date is 14 days before the onset of next menstrual cycle. So at the time of ovulation, this is a Graafian follicle, right? So the secondary oocyte, 14 days before the onset of next menstrual cycle, during ovulation, it releases from the Graafian follicle and it enters into the uterine tube. If the fertilization occurs, then only secondary oocyte completes its meiosis II and converts into mature ovum and releases the second polar body. If the fertilization does not occur, these secondary oocytes shed along with the menstrual blood. That's about the oogenesis. And coming to the structure of the Graafian follicle, this is the secondary oocyte surrounded by the zona pellucida and cumulus ovaricus cells, and these are the granulosa cells. After the release of the secondary oocyte, these granulosa cells, they become enlarged, and they are filled with yellow pigment, and now these granulosa cells we call them as luteal cells, call them as corpus luteum. This corpus luteum plays a very important role, because if the fertilization occurs, this corpus luteum secretes progesterone until placenta starts secretes hormones. We have corpus luteum is of two types: corpus luteum of menstruation and corpus luteum of pregnancy. Corpus luteum of menstruation, the lifespan of corpus luteum is about nothing but 13 to 14 days. And corpus luteum of pregnancy, if the fertilization occurs, corpus luteum of pregnancy starts secreting progesterone until placenta starts secreting hormones. The corpus luteum of pregnancy, the lifespan of corpus luteum of pregnancy is about 3 to 4 months. So that's about the oogenesis. Primordial germ cells into oogonia. Then without cell division, they transform into primary oocyte, and meiosis 1 takes place, and one cell receives major amount of cytoplasm, that is secondary oocyte and first polar body, and the secondary meiosis division takes place if the fertilization occurs, and the secondary oocyte turns into mature ovum and releases the second polar body. That's all about the oogenesis. So coming to the spermatogenesis, it is the process of maturation of the male gametes into spermatids. This is the process of spermatogenesis. Spermatogenesis involves 3 steps. One is spermocytosis and meiosis and spermiogenesis. Spermatocytosis, it involves the primordial germ cells. It carries 44 X5 because it's male, so 44 autosomes, and X5 are the sex chromosomes, and these primordial germ cells, it undergoes mitosis in the seminiferous tubules and gives dark type A spermatogonia, and this dark type A spermatogonia again undergoes mitosis. This dark type A spermatogonia again undergoes mitosis to give light type A and dark type A spermatogonia. So the primordial germ cell, it undergoes mitosis to give dark type A spermatogonia, and this dark type A spermatogonia again undergo mitosis and gives light type A and dark type A spermatogonia. This dark type A spermatogonia again further division, again used for the further division, and this light type A spermatogonia, it also undergoes mitosis and gives type B spermatogonia and this type B spermatogonia again undergo mitosis. And they give rise to primary spermatocytes. So this entire process is called, that means up from dark type A spermatogonia to primary spermatocytes, this entire process is called spermatocytosis. So spermatocytosis, this process is called spermatocytosis. First of all, the primordial germ cells undergo mitosis to give dark type A spermatogonia. Again dark type A spermatogonia again divide to form light type A and dark type A spermatogonia. Light type A spermatogonia again divides to give type B spermatogonia, and type B spermatogonia again undergo mitosis to give primary spermatocytes. These primary spermatocytes now undergo meiosis to give secondary spermatocytes. So these primary spermatocytes now again undergo mitosis to give secondary spermatocytes, and these secondary spermatocytes undergo meiosis to second meiotic division to give rise to spermatids. So this process is called spermatogenesis. It is nothing but the maturation of the male gamete from primordial germ cell to spermatids. This entire process we call it as spermatogenesis. So the male gamete carries 44XY until the female gamete carries 44XX. So when undergo meiosis, when the primary spermatocytes undergo meiosis, each spermatocyte receives, one spermatocyte receive 22X, other spermatocyte receives 22Y. So if the 22Y spermatid combines with the matured ovum, then the 22Y spermatid combines with the matured ovum, the outcome is a boy. If the 22X spermatid combines with the mature ovum, the outcome is a girl. So it depends on the father to decide the sex of the baby. So that's about the spermatogenesis. So here see, dark type A, again dark type A divided into light type A, dark type A, again light type A divided into type B, and the type B undergo mitosis to give primary spermatocytes. These primary spermatocytes again may undergo meiosis 1 to give a haploid number of chromosomes. One cell receives X, other cell receives Y, and these cells again divides into meiosis 2 and divides into X chromosomes. This entire process we call it as spermatogenesis. And when we take the spermatid, so this is the nucleus carrying a haploid number of chromosomes, and this is Golgi apparatus is there, and we have anterior centriole is there, posterior centriole is there when we take the spermatid, structure of spermatid, and we have mitochondria all over the cell, all over the spermatid. Okay, now process of spermiogenesis. The conversion of spermatid to sperm is called spermiogenesis. So spermiogenesis here, see the Golgi apparatus here, they form, the nucleus form head of the sperm. This nucleus forms the head of the sperm, and the Golgi apparatus here, these are the Golgi apparatus. And these are the mitochondria. And these two are centrioles, anterior centriole present. So the conversion of spermatid to mature spermatozoa, this process we call it as spermiogenesis. So here the nucleus forms head of the sperm, and the Golgi apparatus forms acrosomal cap, which covers the two-thirds of the head of the sperm, and from the posterior centriole, axial filaments come from the posterior centriole, axial filaments come, and the mitochondria here, they form the sheath around the axial filaments, and the mitochondria here, they form sheath around the axial filaments, and the axial filament further extend downwards. This is called tail of the sperm. So the nucleus forms the head of the spermatozoa. The nucleus forms the head of the spermatozoa, and the Golgi apparatus forms the acrosomal cap, and the anterior centriole forms the neck, and the posterior centriole, from the posterior centriole, axial filaments extends downwards and they form the body of spermatozoa. And these axial filaments, they are surrounded by sheath. This is called mitochondrial sheath, which is surrounded by the which surrounds the axial filaments. And after that, the axial filaments extends further downwards, and this is called tail of the sperm, and this part is called body of the sperm. This is the head of the spermatozoa. So here the nucleus forms the head, the Golgi apparatus forms the acrosomal cap, and then anterior centriole forms the neck, and the posterior centriole we have axial filaments comes out, and these axial filaments surrounded by the sheath, which is derived from the mitochondria here, and further the axial filament further extends downwards, they form the tail of the spermatozoa. So this process of the conversion of spermatid to spermatozoa, we call it as spermiogenesis. So the entire spermatogenesis consists of 3 steps. First one is spermatocytosis, next one is meiosis, and the third one is spermiogenesis. So spermatocytosis means the primordial germ cells divides into dark type A, and then again into light type and type B, and up to this we call it as spermatocytosis. And the primary spermatocytes divides into secondary spermatocytes by undergoing meiosis 1, and these secondary spermatocytes, they divides again undergo meiosis 2, and they form spermatids. The formation of spermatids to mature spermatozoa, we call this process as spermiogenesis. So that's about the spermatogenesis.